Hi, welcome to Unit 6, Gene Expression Regulation of AP Biology. This is more dense, I'd say, than the last unit, so we are going to do our best to cover it as quickly as possible. So let's go ahead and get started. You have to keep in mind that in the early/mid 1900s, people didn't actually know what the molecular basis of inheritance was. People thought it was DNA, other people thought it was protein. So there are two key experiments to help make the case for DNA, and the first was the Griffith Experiment. So Griffith was studying two strains of Streptococcus pneumoniae. There was a rough strain, which is non-pathogenic, and there was a smooth strain, which was pathogenic. When he gave it to mice, it just killed the mice. So what he found was that if you mix rough strain bacterium with dead smooth strain, it ended up killing the mice. And that's because the DNA from the dead smooth strain was transformed into the prior non-pathogenic rough strain and conferred a new phenotype, the pathogenicity phenotype, and caused the mice to die. So that helped make the case for DNA. And then you have Hershey and Chase. They were studying bacteriophages and bacteria. The phages were grown in, well, what was the whole point of the experiment? Was that the DNA component of the phages entered the bacteria and not the protein component of the phages. But how did they determine that? They grew phages in different media, and they labeled, by that effect, different, they either labeled the DNA of the phages or the protein of the phages. So they grew some phages in radioactive phosphorus, and this ended up labeling the DNA of the viruses. And and you can remember that because DNA incorporates phosphate into the sugar phosphate backbone. Then they also grew some phages in radioactive sulfur, and this labeled the protein of the phages. And you can remember that because proteins require sulfur for the disulfide bonds in the tertiary structure. So after they created these phages in the different media, they infected bacteria. And what they found is when you spun down the bacteria, they only found radioactive phosphorus, which labeled the DNA in the pellet with all the bacteria. And then the supername, which was this media, had all the radioactive sulfur, which was labeling the proteins of the phages. And so this made the case again for DNA. All right, next topic is Chargaff's rule. This is just very simple. It's that your A's and T's are equivalent in number, and your G's and C's are equivalent in number. I'll just keep it simple as that. The context for Chargaff's rule is that people knew about nucleotides, and but they didn't know how it was arranged into a 3D structure. That came later with Watson and Crick. But people are still studying the composition of nucleotides, and that's where Chargaff's rules came from. We'll move on to DNA structure. I'll skip over the rich history behind this discovery of DNA structure and just cut to the chase of what's important about DNA structure. And the first thing you need to know is that DNA is obviously made up of nucleotides, and nucleotides have three basic components. It's the phosphate, the sugar, and the nitrogenous base. So the sugar is kind of the heart of this, so we'll start with that. You need to know how to count the carbons in a sugar. The carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. All right. And then what you have to remember is that for DNA, it's deoxyribose. For RNA, it's ribose. How do you tell the difference? Well, for deoxyribose, the 2' carbon doesn't have like an oxygen, it's just H. And then for ribose, that 2' carbon is OH. The second component is phosphate. That is on the 5' carbon of the sugar. And then you have the nitrogenous base, which is on the 1' carbon of the sugar. What do you have to know about nitrogenous bases? Well, they're split into purines and pyrimidines. There are two mnemonics that help you remember purines and pyrimidines. The first for purines is pure as gold. Purines, adenine, guanine. The second is cut, which is for pyrimidines, and that is cytosine, uracil, and thymine. You should remember that purines are the larger ones. They have 2 rings, while pyrimidines are smaller with a single ring structure. A's and T's go together when they bond with 2 H bonds. G's and C's go together with 3 H bonds. So that's why DNA fragments that are more GC rich require more heat or more energy to separate, because you just have more H bonds keeping the strands together. The 5' end, in terms of directionality of a DNA molecule, the 5' end has phosphate and the 3' end has dihydroxyl. You have to remember that. And DNA strands, when they come together antiparallelly. And the last thing you need to know, which is the most important, is that DNA strands are only extended at the 3' end. Okay? This will help you basically sift through so many of your DNA replication problems. All right, so now that people had figured out the structure of DNA, the next question was, how's it replicating? And that brings us to our next topic, the DNA replication models. So there are 3 of them. We're going to discuss conservative, semi-conservative, dispersive. Conservative, you can kind of think of it as somebody who's conservative, likes to always come back to themselves. So the strands separate. They serve as templates, but then they separate and come back to themselves, the original strands. In the semi-conservative model, you can think of it as somebody who's just a bit less conservative and they do a bit more mixing. So the strands separate, they serve as templates, but then they stay with their newly synthesized strands. And then the dispersive model is the most, dispersive and random. Each daughter molecule gets a bit of each, parent molecule in the dispersive model. Out of these 3, obviously the correct one is the semi-conservative model. I will say you should probably know the first and second replication tube patterns for these 3 different models. I would work through them, but just for the sake of time, I'll have to keep going, and we'll veer into DNA replication, which is the next topic. So for DNA replication, people are like, okay, here's the structure of DNA. This is the model that we think it's replicating in. Well, how's it actually happening? Well, I'll split into the replication fork, like all of the players and actors in the replication fork, and then into the leading strand synthesis and lagging strand synthesis. So at the replication fork, you have helicase, that is unwinding the DNA. You have SSB, that is stabilizing unbound DNA. You have primase, which actually lays down your RNA primer, from which DNA polymerase can bind and extend. And then lastly, you have topoisomerase. And topoisomerase essentially creates these nicks in the DNA to relieve strain and detangle DNA during replication. Okay, so how does the leading strand get synthesized? So for the leading strand, DNA polymerase will bind to the primer and will start extending off of that RNA primer with DNA nucleotides at the 3' end. And for the leading strand, it's simple. Like as that replication fork opens, the DNA polymerase is replicating into the replication fork. So as it opens more, the DNA strand can just keep on extending more and more, because each time they're adding at the 3' end, it's just extending into the fork. In eukaryotes, we have 11 different DNA polymerases, and they lay down 50 base pairs per second. So it's insanely fast. And that's all you need to know about the leading strand. The lagging strand is more complicated. That's where you form the Okazaki fragments. So for the lagging strand, we'll switch over to E. coli. Primase lays down the primer. DNA polymerase 3 will extend that primer at the 3' end until it hits another primer. Then DNA polymerase 1 has to come and replace those RNA nucleotides in the primers with DNA nucleotides, but then you still have these nicks, and that's where ligase comes. And ligase will come and seal the nicks in the sugar phosphate backbone. And that's how you synthesize off of the lagging strand. But just keep in mind, as the replication fork opens for the lagging strand, the DNA polymerase is synthesizing away from the fork. And then as it opens up more, it'll hit another primer and replicate away from the fork. Okay, so that is how DNA replicates. The next topic is the central dogma. And what the central dogma basically tells us is that you have DNA that gets transcribed into RNA and RNA that gets translated into protein. It's a very central, simple concept, but very encompassing. So we're going to move on to the middle part of the central dogma, RNA. and how you get there, which is via transcription. So we've done DNA and DNA replication, but now the question is, how do you express genes in DNA? Well, you do that via a process called transcription. In prokaryotes, they have a single RNA polymerase, and in eukaryotes, we have 3. And the RNA polymerase that synthesizes mRNA is called RNA polymerase 2. RNA polymerase does not require a primer to bind to a region in the DNA called the promoter. So there are 3 steps, though. You have initiation, elongation, and termination. So for initiation in eukaryotes, you have transcription factors that bind to the promoter, and there's a region in the promoter called the Tata box. And so these transcription factors bind, and then RNA polymerase binds and can initiate transcription. For prokaryotes, it's more simple. The RNA polymerase can bind straight to the promoter and initiate transcription. For elongation, RNA is always extended at the 3' end. Just as for DNA replication, DNA is always extended at the 3' end. Same for mRNA, always extended at the 3' end. It also proceeds at a very fast rate. In eukaryotes, it's like 40 nucleotides per second. And what's important to keep in mind is for a single gene being transcribed, you can have multiple RNA polymerases all running the same gene, and they'll synthesize mRNA at the same time simultaneously, and the length of the mRNA molecule coming off of the gene will be an indication of how long that RNA polymerase has been transcribing. In the last step, which is termination, in prokaryotes there's a terminator sequence in the DNA. Once the terminator sequence gets transcribed, the mRNA detaches and it's ready for translation. In eukaryotes, we have something called a polyadenylation signal, which is AAU, AAA. And that sequence, once it gets transcribed, you have proteins that come, they cut the mRNA, and then it's ready for pre-mRNA processing. Okay? And that's all you need to know for transcription. Once you have your gene transcribed into an mRNA molecule, you need to do processing, which is our next topic. For RNA processing, the 5' end gets a guanine cap. The 3' end gets your poly A tail. These are important for protecting the mRNA from being degraded, being degraded. So 5' end, guanine cap, 3' end gets poly A tail, and then you have introns that get excised out by spliceosomes. And spliceosomes are a complex of RNA and proteins. And those RNAs are called small nuclear RNAs, snRNAs. You can also have RNAs that can self-splice without the need for proteins, and those are called ribozymes. And that's because they're RNA molecules with enzymatic functionality. Great, so now you have your processed mRNA molecule. Now you need to translate that, right, into a protein. That's the last part of the central dogma. So to understand translation, you need to understand a few things before that, and that'll be our next topic, codons and tRNAs. You have 20 amino acids and 64 codons. Each codon is made up of 3 nucleotides. So you get the 64 because you have 3, you have 3 nucleotides and 4 nucleotide options per base. So it's 4 times 4 times 4, it gives you 64. Out of these 64 codons, 3 are stop codons. Another mnemonic to help you remember the stop codons is you are annoying, you go away, you are gone. These are your 3 stop codons, UAA, UGA, and UAG. You should keep in mind multiple codons can encode the same amino acid, and they typically differ in the 3rd position, which is called the wobble. And the last thing is that the start codon is AUG. So that's codons. What about tRNAs? tRNAs are single-stranded RNA molecules. They fold into this L shape. They have a region, an anticodon region that binds to a codon in the mRNA. And then at the 3' end, they have an amino acid attachment site. You have these 20 amino acyl tRNA synthetases. And these 20 amino acyl tRNA synthetases, they bind to a tRNA, and then they bind to amino acid and attach the appropriate amino acid for each tRNA. That's what they do, and it's an ATP dependent process. Don't forget that. All right, so you know about tRNAs, you know about codons. The last thing is ribosomes, and then we can tackle translation. Ribosomes, which is the next topic, They're interesting. They're obviously made of RNA and protein, but by mass, they're actually primarily RNA. 2/3 RNA by mass, 1/3 protein. You have a small subunit and a large subunit, and they obviously differ in between eukaryotes and prokaryotes, and that's important within the context of antibiotics, but we're going to skip over that now for time's sake. All right, so now let's tackle translation, how you go from mRNA to protein, the last part of the central dogma. Translation has initiation, elongation, and termination. In initiation, you expend 1 GTP. In elongation, you expend 2 GTP per amino acid added, and then you just have termination, which doesn't require any GTP. All right, so how does initiation occur? So in eukaryotes, the small subunit will bind to the initiator tRNA, and then that will bind to the 5' guanine cap and just start scanning the mRNA molecule 5' to 3'. When the initiator tRNA/small ribosomal subunit finds that AUG start codon, it essentially clicks, expends the GTP, and then the large subunit will bind. This then forms what we call the translation initiation complex, and now you're ready for elongation. Elongation has will split into 3 steps. You have to recognize a codon, you have to form a peptide bond, and then you have to translocate. But what do you have to know for elongation? You should know that the ribosome has 3 sites. EPA, you can remember that like Environmental Protection Agency. Each letter represents, something. I like to remember E as for exit, P for protein, and A for arrival. Okay, so remember, small ribosomal subunit is reading, or the whole ribosome is reading the mRNA 5' to 3', 1 codon at a time. Step 1 is codon recognition. You have your existing tRNA in the P site with your existing, just say, polypeptide, and then you have A site that's empty. A new tRNA comes into the A site, and it recognizes a codon there. And for successful codon recognition, you need to expend a GTP. Okay, that's step 1. Once the new tRNA has arrived in the A site and recognized the codon, which uses a GTP, second step is peptide bond formation. What you have to remember is that peptides, or proteins, they have a N-terminus with an amino group and a C-terminus with a carboxyl group. And proteins are always extended at the C-terminus. Okay, so during peptide bond formation, which is step 2 of the elongation phase, what happens is that peptide on the tRNA in the P site will form at the C end, C-terminus, will form a peptide bond with the amino acid on the tRNA in the A site. So the whole peptide shifts from the tRNA in the P site to the tRNA in the A site. And that's step 2. And step 3 is translocation. The ribosome expends another GTP to essentially pull the mRNA molecule in and through the ribosome. And in this step, essentially everything translocates. So now the empty tRNA in the P site ends up in the E site and gets ejected, and then the amino acid in the A site that has the whole peptide will end up in the P site. And now you can repeat the process. Okay, termination. You remember you have your 3 stop codons. You are annoying. You go away. You are gone. And once a ribosome reaches the stop codon, a protein called the release factor binds. It promotes the addition of a water molecule that detaches the polypeptide from the tRNA, and it's free. And that's it. That's the end of translation. Remember how you can have multiple RNA polymerases that run through a single gene? You can also have multiple ribosomes translating a single mRNA molecule, and those are called polyribosomes. Okay, let's talk about mutations now. So you can have single base pair mutations in DNA that cause changes in proteins, like that we just discussed. So single base pair substitutions are classified into 3 classes. They can be silent, missense, or nonsense. For silent mutations, the DNA base pair change doesn't actually result in a change in the amino acid, right? Because it's a different codon, but that codon also encodes for the same amino acid. For A missense mutation, the DNA base pair change, results in a different codon and subsequently a different different amino acid. That's obviously very deleterious, especially like in the context of sickle cell anemia. Then third, you have nonsense. In a nonsense mutation, the single base pair substitution results in a premature stop codon. That's also pretty risky because you essentially just stop producing that protein in the cell. Also keep in mind that insertions and deletions can also be very deleterious because it can result in a frame shift mutation, and when you shift the frame, you're just reading the entire mRNA wrong because you're off by a base pair or two, and you end up having multiple missense mutations, multiple different changes in amino acids, and that can be very deleterious as well. I'll stop there, but if you're interested in how to target mutations in cancer and form treatments that only kill cancer cells and not healthy cells, I would encourage you to look at adoptive cellular therapies in cancer immunotherapy. All right, let's move ahead on to the next topic, which is gene regulation. So for gene regulation, we'll cover in prokaryotes and eukaryotes. So in prokaryotes, we're going to have to discuss the lac operon, and I'll try to keep it as simple as possible. So an operon, what is an operon composed of? It's composed of a promoter, which is a segment of DNA to which the RNA polymerase binds. Then you have the operator, which is a segment of DNA to which regulatory proteins bind. And then you have the actual genes whose expression you're trying to regulate. That forms an operon. So the lac operon, what's going on intuitively? When lactose is present, you want to transcribe lactose metabolism genes so you can extract energy from lactose in the environment, right? Simple enough. But that process needs to be positively and negatively regulated. And that's what we're going to discuss through 3 different scenarios. The first is a no lactose scenario. When there's no lactose in the environment, allolactose is low. It does not bind to the repressor protein, and the repressor protein can bind to the operator and block transcription of the lactose metabolism genes, which makes sense because why would you want to make them when there's no lactose in the environment? Okay. What about a high lactose scenario? In a high lactose scenario, allolactose is high, and it binds to the repressor protein, changes its shape, and it can now no longer bind to the operator. This allows RNA polymerase to bind to the promoter and transcribe lactose metabolism genes, which it will do at a low basal rate. For the genes to be transcribed in larger amounts, you need positive regulation, which is the third scenario. In positive regulation, the third scenario is when you have high lactose and low glucose. So again, high lactose, high allolactose binds to the repressor. Repressor can't bind to the operator. RNA polymerase transcribes at a low basal rate. Then there's also no glucose in the environment, so the cell really needs energy. And in that case, when glucose is low, cyclic AMP is high, and that binds to a positive regulatory protein called CAP. And then CAP can bind to the promoter and positively regulate and promote transcription of the lactose metabolism genes at a much, much higher rate. And we'll end there for gene regulation in prokaryotes. Those are the 3 scenarios for the lac operon. Okay, what about gene regulation in eukaryotes? It's more complex, and I'll break it down to the core components and the essential points for each one, but we won't have time to go into depth for each. Okay, first concept you have to keep in mind is that when your father's sperm and mother's egg fuse together, fertilize into a single diploid zygote cell, that diploid zygote cell has primarily undergone only one process to give you today, and that's mitosis. And in mitosis, you essentially just replicate all of your DNA into 2 daughter cells, right? So what that means is that all of the cells in your body essentially have the same DNA sequence, by and large. But then what makes that cell in your eye different than the cell in your toe? And the answer is you have the same DNA in those 2 cells, but it's the differential expression of the genes in those 2 cells that result in the diversity of cells that you see today in your body. Okay, so you have about 3 billion nucleotides in your genome, and out of those 3 billion nucleotides, you have about 20,000 genes, and it's those 20,000 genes differential expressions that lead to different cell types. Okay, what are the modification classes when it comes to modifying DNA? So first is histone modifications. You can acetylate histones, and this generally loosens DNA into euchromatin and promotes DNA expression. You can deacetylate histones, and this will generally compact DNA into heterochromatin, and this will generally suppress DNA expression. Okay. Second is DNA modifications. You can methylate DNA. When you do so at promoter sites, it generally silences genes. Another thing to keep in mind about methylation is that methylation patterns are inherited to daughter cells. You can do transcriptional regulation. You can have activators and repressors that bind to enhancer elements upstream or downstream of the gene of interest that will either promote or suppress transcription respectively. You have post-transcription regulation. mRNA lifetime and degradation can be modulated by nucleotide sequences in that 3' UTR, the 3' untranslated region. You can have post-translational regulation. You can ubiquinate proteins and mark them for degradation in proteosomes. And then lastly, you can have non-coding RNAs, which are microRNAs that combine with proteins, and then they combine, targets with complementary sequences, and they can block mRNA from being transcribed, or they can just degrade mRNA directly. All right, and then we'll finish off with biotechnology. We'll cover some basic topics in biotechnology and the purpose and function of each. So for PCR, polymerase chain reaction, the whole point there is just to amplify DNA. You do this with obviously having your DNA polymerase, which will add bases. You need your actual bases, your ATs, Gs, and Cs. You need ATP as an energy source. You need primers. So the DNA polymerase can have a site to start at, and then you need your DNA template. What's unique about what's unique about PCR is that you don't need much DNA to do PCR. I've done PCR from just a single cell, and I can amplify a particular locus like the TCR locus in that single cell, and you can get a lot of DNA from it. All right, second is bacterial transformation. The whole point here is to introduce foreign DNA into a cell. You can do that through heat shock. The sudden increase in temperature will create pores in the plasma membrane and allow foreign, material like DNA to enter into the cell. That's transformation. Third is gel electrophoresis. This allows one to separate molecules based on size and charge. In gel electrophoresis, DNA is generally negatively charged due to the sugar phosphate backbone, and it travels towards the positive anode, and smaller molecules will travel farther. And I'll end with DNA sequencing. We'll talk about Sanger sequencing. So for Sanger sequencing, you basically have your DNA, template or a strand that you want to sequence. And what you do is you add DNA polymerase, you add your normal, deoxyribonucleotides, your ATs, Gs, and Cs that are just normal and unlabeled. You add your energy source, like ATP, and then you add these fluorescently labeled dideoxyribonucleotides. They're dideoxyribonucleotides, which means they lack an OH at that 3' carbon, so once it gets incorporated, DNA polymerase can no further extend that DNA strand because there's no OH for it to, add another nucleotide. So what does this mean? This means statistically, when you add all these together, each base pair position on that DNA template strand will eventually incorporate a dideoxyribonucleotide, and it'll terminate replication at that point. These dideoxyribonucleotides are also fluorescently labeled. So what that ends up meaning is you have all these different DNA strands that differ in length by one base pair because they all terminate with a dideoxyribonucleotide, and they fluoresce a specific color based off of whether they terminated with an A, T, G, or C. Last thing in Sanger sequencing is that you can now separate these DNA fragments in a gel column, and you can separate DNA fragments that differ in length by one base pair. So the smallest fragment will maybe fluoresce a particular color for G, and then the next fragment that is one base pair longer will fluoresce a particular color for A, and et cetera, and that'll give you your, it'll give you a DNA sequence of A, T's, G's, and C's, from which you can infer the DNA sequence of the original template strand. And that's Sanger sequencing. And that's all for Unit 6, Gene Expression Regulation of AP Biology. So we started off with the two experiments that helped make the case for DNA being the molecular basis of inheritance, which was the Griffith Experiment, which looked at the rough and smooth strains of Streptococcus pneumoniae, and the Hershey and Chase experiment that looked at radioactively labeled phages and with bacteria. And then we looked at Chargaff's rule. It basically tells you A's equals T's and G's equals C's. And then we looked at DNA structure, and we talked about how it's three main basic components: the sugar, phosphate, and nitrogenous base. 5' end with phosphate, 3' end with hydroxyl, and DNA is only extended at the 3' end. We looked at the three different DNA replication models: the conservative, semi-conservative, and dispersive, with the semi-conservative being correct. And then we looked at DNA replication. How do you make more DNA from DNA? Looking at the leading strand and lagging strand. Then we looked at the central dogma, which is that DNA goes to RNA, which then goes to protein. And we looked at RNA transcription, which had initiation, elongation, and termination. And for RNA, for transcription initiation, you have the Tata box and the promoter in eukaryotes. And then we looked at RNA processing. 5' end gets the guanine cap, 3' end gets the poly A tail, and then introns are excised out by spliceosomes. We then discussed codons and tRNAs. The three stop codons are you are annoying, you go away, and you are gone. UAA, UGA, UAG, and the start codon is AUG. We then looked at, we talked about ribosomes, and then translation, where you use 1 GTP in initiation and then 2 GTP in elongation per amino acid added, and then you have termination with the release factor protein that binds to one of the stop codons. Then we discussed the three classes of mutations: single base pair mutations like silent mutations, missense mutations where the amino acid changes, and nonsense mutations where you have a premature stop codon. Then we discussed gene regulation prokaryotes with the lac operon and the three different cases. You have your no lactose scenario, your high lactose scenario, and your high lactose and low glucose scenario, where no lactose was negative regulation and high lactose and low glucose was your positive regulation with CAP. And then we discussed gene regulation eukaryotes, where you can acetylate histones for expression, gene expression, and deacetylate generally for suppression, DNA methylation and promoter sites, transcriptional regulation with activators and repressors, post-transcriptional regulation with 3' UTR sequences, post-translational regulation with ubiquination, and non-coding RNAs that can degrade or block mRNA. And then we finish off with biotechnology, discussing PCR, amplifying DNA, transformation, where you're introducing foreign DNA into a cell, gel electrophoresis, where you have negatively charged DNA traveling towards a positive anode, and then DNA sequencing, Sanger sequencing, where you essentially use these fluorescently labeled dideoxyribonucleotides and then run them through a gel column to determine DNA sequence. That's all for Unit 6, Gene Expression Regulation. I hope you found that helpful.